Well, welcome everybody to HydroTerra's latest webinar. This week, the topic is antimicrobial resistance as an emerging concern. Is our wastewater treatment enough? I'd like to welcome back Brent Davey, this would be his second or third webinar, and really appreciate your contribution, Brent. Brent is a principal environmental scientist with Fife, and I'm the managing director of HydroTerra. Before we get started, I would like to begin by acknowledging that we conduct our work across this great land, and for that privilege, we would like to thank the traditional owners. HydroTerra respectfully acknowledges the Boonwurrung people of the Kulin Nation, where we are located today, and we pay our respects to their elders, past and present. There's a picture of Dr. Brent Davey. When I first met Brent, he used to go around in a bow tie, so he's obviously getting a bit more casual with his clothes as he gets a bit older. It's good to see you, Brent.
Thank you, Richard.
A little bit about our presenter. So Brent Davey has a Bachelor of Science and Honours, a PhD, and is a certified environmental practitioner. He has worked in land remediation and contamination management for over 45 years. He began his career as a molecular microbiologist, interested in how microbes become resistant to antibiotics and chemical toxins, which in turn led to work in biotechnology in the development of wastewater treatment systems, bioremediation, and the assessment and remediation of site contamination. His current practice includes the management and removal of PFAS from soil and water, acid sulfate soil management, the potential and limits of analytical chemistry for the site contamination industry, and emergent contaminants, and whether nature can save us from ourselves. We love your questions, and thanks so much for all the early bird questions that people have sent in, some really good ones there. If you would like to raise a question during the webinar, please use the Q&A button at the top of your screens, and we will do our best to answer those questions at the end of the presentation. Why does HydroTerra do these presentations? Well, we like to share knowledge, and I think the industry needs to share more knowledge. We like to facilitate education, and we like to take an industry leadership position. So without further ado, I will hand over to you, Brent.
Thanks, Richard. So before we start, it's worth just answering the question, what are antimicrobial agents? These are antibiotics, as we know them commonly. And this was sort of a fun fact slide. Antibiotics are credited with having saved millions of lives over the last 100 years, which is largely the period that we've had access to these substances. WHO argues that the use of antibiotics has extended the average human lifespan by 23 years, which is extraordinary when you think about it. The other fun fact is that at least 1 in 3 Australians have been prescribed an antimicrobial agent, an antibiotic, at least once in their lifetime. It may well be more, but even 1 in 3 puts us in a fairly advanced league in terms of antibiotic use per head of population. And what is antimicrobial resistance? It's the ability of organisms which used to be susceptible to these agents to resist the inhibitory or lethal effect of those chemicals. And WHO estimates that by 2050, unless we manage to find some way around it, antimicrobial resistance will cause up to 10 million deaths worldwide. Add $1 trillion to healthcare costs, and perhaps more importantly, render many medical and surgical procedures impossible. This is certainly something worth thinking about. In Australia, various Australian sources suggest that AMR, antimicrobial resistance, causes more than 1,600 deaths per year, and overall could wipe the best part of nearly 300 billion from the economy by 2050. So it's a sobering thought and clearly a potential problem.
Going on with that picture there, Brent.
This is a gentleman who, before the age of antibiotics, he's a surgeon, and he's about to undertake an amputation. But unfortunately, his success rate is about 50-50 in this particular instance, which meant that basically 1 in 2 of his patients died of infection as a result of the surgery. And it's been improvements to that extent that antimicrobial agents have allowed. But that doesn't happen anymore. But that's, I think, 1830 or thereabouts. So what causes antimicrobial resistance? Basically the overuse or the misuse, and surprisingly or paradoxically, the underuse of antimicrobial agents. Very often we get prescribed antibiotics for conditions that will resolve themselves in a few days. Flu, diarrheas, those sorts of things. They really don't need antibiotics, but our medical professions are so stressed that very often, here, take this pill, is the response, and we've sort of bought into that. But if you have been prescribed an antibiotic, very often people take the pills until such time as they feel better. Whereas in fact, what they need to do is take the recommended complete course. And what happens, of course, is that if you don't take enough, there will be organisms that survive. Perhaps you will no longer be ill because you've developed some degree of immunity, but the organisms that survive are now more resistant than the ones that began. Very often too, the agent prescribed is very broad spectrum because you don't go to the doctor with an infectious disease and a document that says, I have this particular disease. Very often, the response of the medical profession has to be, here, I'll give you something broad spectrum because I'm not quite sure what is causing your problem, but this should fix it. Whereas if we could prescribe only an agent that is known to be specific to the particular illness that you have, the outcome would be much less likely to produce resistant organisms. That's a whole lecture in itself, so you'll have to bear with me if you don't understand the ins and outs of the activity of different antibiotic agents, but it's certainly a cause where broad spectrum antibiotics are used when more narrow spectrum would be more appropriate. And the other one, which fortunately doesn't happen much in Australia, is where antibiotics used for human therapy or in human medicine are also used to treat diseases in veterinary practice and agriculture. This might sound like a logical thing to do, but in fact, because there are so many animals in across the country, it could itself cause real problems if clinical antibiotics are used and not preserved only for human work. Fortunately, there are regulations that mean that the veterinary and agricultural antimicrobial agents are quite different from those used clinically in most cases. So we don't have situations as, say, the Americans do, where things like piggeries and feedlots are a major source of resistant organisms that affect us. But it's not all about us. The natural processes in soil and water are really the main source of antimicrobial resistance. And I became interested in this area, particularly, given my background in extrachromosomal genetics and antibiotic resistance, when I realized that very often it was contaminants like cadmium, mercury, even zinc that actually selected antibiotic resistant organisms in soil and kept them emerging. And it's important to remember that most of our antibiotics originally came from soil microbes. So the soil already has a background of antimicrobial agents in it. And it's that antimicrobial soup, if you like, that is constantly acting to pressure the microbes that live in soil and select for organisms that are more resistant to them. And so wastewater in particular is a source of both new and continuing pressure on the bugs in soil because it keeps introducing new stressing chemicals into that environment. Next one, Rich.
Just before we do, so this correlation between contaminants like zinc and antimicrobial resistance, just can't quite get my head around why that happens.
Okay. What we don't realise is that one of the reasons zinc is on the list of contaminants is that it actually is more toxic to other critters apart from mammals. So it's quite a plant toxin, but it is also toxic to various microbes, bacteria and fungi that live in the soil. So if you've got soils that are rich in available zinc, and very often that's what happens when you've got lots of runoff from galvanized iron and that sort of stuff in an urban environment, or from tyres on roads and those sorts of things. It's the zinc that actually inhibits all these bugs that are already in soil. They don't. It's something that slows them down or even kills them. Now, that then provides a selective pressure where if you are more resistant to zinc than your neighbours, you prosper in this soil. And it turns out that there are the mechanisms by which bugs are resistant to zinc are often linked to genes that provide resistance to other things, including antibiotics.
Okay, makes sense.
And so really soil is where it's all happening. The bugs live in soil. You've got a whole heap of bacteria, fungi. You've even got viruses, free viruses, but they actually parasitize the other organisms that are in there. You have soil nematodes, tiny little worms. that are hosts to bacteria and fungi and so on. And then all the organisms that live in the root zone associated with plants. Now, this whole environment is awash with genes, with DNA that confers resistance to all sorts of antimicrobial agents, including the antimicrobials that microbes in the soil themselves produce, be they antibiotics, or there are other agents that various microbes produce that enhance their competitiveness in that soil environment. So one of the other things is that there are chemicals known that actually switch on the ability, a microbe may carry a gene but not express it. And so there will be substances in the soil that switch that gene on that allow the organism to compete better in the soil. And so we can consider soil as basically a Darwinian microbial environment where really survival of the fittest is what is driving this whole thing. And the question becomes, where do these stresses and antimicrobial agents come from in the soil? I mentioned that some microbes themselves produce them, but the great source of these days is now recognised to be wastewater.
Just before we move on, Brent, so how quickly does this happen, right? So you've got a bacteria in the soil. I mean, how frequently is it replicating? How quickly do you sort of change from a normal sort of microbial population to a AMR style microbial world?
If the conditions are right, and this is what much of laboratory microbiology is about, your organisms can double in 30 minutes. And that's one of the aspects of microbiology that makes it a threat to us and environment or whatever, that this process can be very fast. That if there is a selective agent applied to soil or a wastewater body or whatever, the change in microbial population can be literally overnight. So it can happen very fast. So this is a summary diagram of that process and our role and association with it. I need to explain a couple of terms here. The first is microbiome. This is a fancy name, I guess, for the entire zoo of microorganisms in soil, and considers it as a whole, as an entity of itself, because it's all interlinked. So you've got all these soil microbes of different sorts, but they're all there in the soil. Their genome, their genetic material, therefore becomes the metagenome. And it's from that metagenome that turns out that all these resistance genes we're talking about can be developed and taken and adopted by organisms to make multiple resistant ones. So what happens is that process goes on as a stressor is applied to the soil, and is basically allowed to develop, and the whole thing is that you've got a situation where the pressure on the organisms is such that genes are adopted, you get a resistant organism that has picked up a piece of DNA conferring that resistance. And then we come along as workers working in soil, and that organism can infect us. I should stress that infection doesn't necessarily lead to any overt symptoms or disease, whatever. If we're healthy and strong, we'll normally have these organisms become established in our skin, possibly even in our digestive system, without causing any disease whatever. But we then go home and we can pass these organisms on to our family members and to our pets. And they too may not get sick. But if there is someone amongst them that is immunocompromised, which may mean just old, or a very young baby, or someone with open wounds, or at least surgical or wounded open skin or whatever, the net effect can be that that person may then suffer from what I have labelled here as MDR illness, multiply drug-resistant illness. In other words, an illness with antimicrobial resistance. And the one that really worries us is where the resistance is to several different antimicrobial agents. Okay, so therein is the problem in terms of its effect on us. But then, of course, we all contribute to sewage and our wastes, and that one way or another winds up in wastewater. And so you've got this concept of the microbiome of wastewater, and those organisms floating around in wastewater also have DNA that can be considered as sort of one big genome, and that genome carries various resistance genes. So the whole circle keeps going round and round. And the important thing is that, as you'll see on the next slide, that the different processes happen in all of these media, both in the soil and in the wastewater. So what you've got is, and this slide focuses on soil, but in soil you've got a few known non-pathogens. In fact, you've got quite a few. You've got, and a pathogen is an organism that makes you sick. Most organisms in soil don't do that. They're quite harmless to people or to animals. But there are some that are pathogens, that's organisms that are capable of making you sick. Most of those are a special class called spore formers, but that's just their way of surviving in the soil. There are others that would not normally make you sick unless there's something in you that predisposes you to that, immunocompromised or whatever, and they're called opportunistic pathogens. But they account for less than 10% of the microbes that are present in soil. One of the difficulties that soil microbiologists have even today is that most of the organisms in soil are very difficult to cultivate and isolate by traditional microbiological means, petri dishes and that sort of stuff. But they're there because we know that we can detect gene sequences from these organisms in the soil. And the way that happens is that as cells die for whatever reason, whether it's due to soil constituents and antimicrobials in the soil, like sort of chemicals, or the action of bacterial viruses, there are a lot of those in soil as well. When the cell dies, for whatever reason, its DNA is liberated into the soil, and that becomes your soil metagenome. And it can either be fragments of the one big chromosome that most single-celled organisms, such as bacteria, have, or it can be a structure called a plasmid, which is a small loop of DNA, considerably smaller than the what you'd call a chromosome of a bacterium or a fungus. And it's usually plasmids that carry the genes for various resistance. And so all of that nucleic acid, all of that DNA is present in the soil as sort of a soup. And microbes are in constant contact with this. And there are now known to be 3 processes whereby the DNA conferring various capabilities can be acquired by an organism. The first is simply is a process called transformation, where simply a piece of DNA sticks to the outside of a microbial cell. There are processes that have evolved that allow that cell to take the DNA intact inside, and once inside the cell has evolved machinery that says, is this DNA any use to me? Often it is just to repair ordinary living functions of that organism. But it can also say, oh, wacko, here is a gene that allows me to be more resistant to zinc, which is a lot of it in my environment, or to penicillin or whatever. And that can then be incorporated into the genome, the genetic material of that particular organism. I mentioned earlier over here that bacterial viruses cause the lysis and death of bacteria, but they, in the process, can often pick up pieces of bacterial DNA themselves, and that then can be injected into another target cell that the virus targets in a process called transduction. You get little pieces of DNA that, again, get introduced into the cell, and the cell can decide, is this DNA useful to me? And one of the earliest processes by which genes could be transferred between organisms, if you like, bacterial sex, was direct contact between cells where the nucleic acid was transferred down what is called the sex pilus, which is a tiny tube linking the 2 bacterial cells. And that's a process called conjugation. Now, these 3 processes are ways by which previously susceptible organisms can become resistant to various agents, including antibiotics, but also other things. And since those other things can include genes that make a particular organism fitter at surviving in a particular environment, what often happens is that is what comes first, but it drags with it some genes for antimicrobial resistance. So we have a system where it doesn't have to be antimicrobial agents exclusively that cause new combinations of resistance to evolve. And it keeps going on, and cells simply respond to what is the current stressor in their environment.
So, Brent, this wacko moment where it decides whether or not it wants this DNA that's conferring this AMR gene, is it more like a hoarding process? So it's just lucky and this gene comes along, or does it actively push those unwanted bits of genetic material out? I'm sort of struggling to believe it's that sophisticated.
It is quite sophisticated, but it's also a lottery, if you like. But it's a lottery in which things like 1 in a million or 1 in 100 million or even 1 in 100 billion is no trouble at all. You only need one cell out of a population of a gazillions in this soil environment to decide, or whether it's an active process or whatever, that it will become resistant. That one cell is then enough to multiply, and as you talked about, overnight can now become a very significant population of resistant cells. So it's a numbers game in that sense. And most of the DNA that's ingested, of course, in this process or transferred by whichever mechanism is discarded and becomes a nutrient in the cell rather than anything else. But it does cause resistant populations as well.
Back in the day, we used to think it was mutation, and that mutation was like a DNA replication problem.
Yep.
But as the world changed, has it, and we now think these other processes are producing that variability.
Mutation still occurs, and it still occurs at the sort of frequency it used to. 1 in a million or 10 million or so. But microbes we've learned that microbes are actually much more adept at adopting sort of existing, why wait for a mutation to happen when you can actually go down to the hardware store and get the whole kit and caboodle in one hit, which is essentially what's happening in soil, that there are whole genetic structures that give you fully functional and formed capabilities without having to wait for a mutation to occur. And it's wastewater that then disperses both the antimicrobial resistant organisms, but it also disperses the selective agents that drive that as well. So I mentioned zinc, that's only one of them. There's a whole heap of others. But the vector, if you like, since soil doesn't move so much, what drives and changes the selectors and stressing agents in soil is wastewater. And so if there are shortcomings in our wastewater management, that's where AMR will tend to emerge and be spread, and where we're spreading both the organisms and the genes that confer that resistance. So this is a diagram that is trying to explain the centrality of wastewater in this process, whereby, the wastes from those three groups of humans that I talked about earlier, healthy workers like us, our family members, and then patients with actual disease that's resistant to antimicrobes, microbial action. Our wastes go either into municipal sewage, or if we're not in areas that are served by sewage, into septic tanks. In addition, our pets and livestock, where they are treated with antimicrobial agents, their wastes lead to are deposited and wind up in stormwater one way or another. And the net effect is that various populations, if you like, of potential pathogens with resistance and nucleic acids, DNA, with carrying genes for antimicrobial resistance, are sort of swept into soil. And so you've got what you might call a hardware store in the soil, that's where all these things are delivered, and these processes go on, where a bug can find predetermined genes for resistance. And the point of this slide is that wastewater treatment plants are reasonably effective at preventing pathogenic organisms, or even most organisms, from being carried through into the effluent they discharge. But we have very little idea about how much DNA is in that same effluent. But we do know that there's lots there. And this winds up in the soil, and this is where organisms can acquire new genes. So wastewater is constantly delivering the antimicrobials and the selective agents into this environment. Septic tanks, as I point out, are less effective at preventing active whole organisms from entering this environment. So it does happen. It just happens sort of that they are more effective at that than certainly wastewater treatment plants. It's not much of a problem. So the next slide.
Just before we do, so wastewater treatment plant obviously has a whole lot of chemicals in it to bust up those microbes.
Yep.
Do you get the same sort of selection pressure though that you're talking about, where like zinc was causing flavoring of the AMR because the others couldn't cope with it? Are we sort of creating another environment that's preferential for it?
Yeah, you will. But the whole industry of wastewater treatment plants, much of it has developed using technology which actually breaks down microbial cells. You know, the whole treatment process, oxygen is actually very good at breaking organisms apart. The kind of maceration that is usually done in wastewater treatment plants also tends to break things down. And over time, the vast majority of microbes in sewage do break down. They're broken down by various things. But the survivors, the few that are left, have access to all these different genes, and in the presence of various chemical stresses, which don't get broken down as fast in a sewage treatment plant, they will become resistant to those agents. We know that a lot of antimicrobial agents will travel right through wastewater treatment plants, often with a substantial reduction, but there's still some left at the end. And this is one of the challenges that wastewater treatment industry faces, that it's the chemicals in the wastewater and sewage that are as much of a problem as anything else. How to break them down and make sure that they don't have an adverse effect on the environment. And if you want an adverse effect on the environment, it is that providing a stressor that selects for resistant organisms in soil.
I'm just thinking about a project we did monitoring mountains of biosolids, and heaps of bacterial activity in those, and they've obviously been right through a sequence.
Yep. Yep.
So it's not like there's a shortage of bacteria at the end of it, or microbes. There's literally tons of it.
I'm trying to be as generous to the industry as I can, because they are challenged with an enormous problem, really. They do get rid of most of the pathogens, but there are certainly lots of microbes left, as you say, in biosolids and those sorts of things. And perhaps one of the important things about my presentation is that the organisms don't have to be ones that cause us disease to actually be creating a problem. They're the ones in which the genetic mechanisms go on. You try next slides we can get to that. um, these are some of the sources of AMR in soil. It's fairly straightforward. You know,
You want to
bush.
talk to this in detail, Brent?
um, but the thing is that modern sewage treatment plants are generally less of a problem, but even then we don't know much about how much DNA, particularly AMR DNA, is transferred right through these systems and discharged. And as you pointed out, there's lots of bugs in, that survive in the biosolids, etc. And inevitably some of those will be transferred in the liquid effluent as well.
It's the same gene process, less sort of transference occur in water, or is it
Yes.
mainly in soil?
I mean, the evidence seems to suggest that it can happen in water. In fact, most of the experiments I used to do, conjugation experiments, you would do in an aqueous environment, in a broth. But we've also learnt that any sort of surface, biofilm, the surface of particles or whatever, is even better at actually promoting this genetic transfer between organisms. And that's because of surface processes helping to, to anchor the DNA so that bugs can process it one way or another.
Yeah.
And in thinking about all of this, there are some myths that sort of at least started when we started to wonder what was going on with the development of resistant organisms. One of the first, and it was sort of a Darwinian argument, was that microbes carrying resistance genes would be less fit and would sort of disappear in the absence of the resistance. Basically the idea was that the selector would be necessary to keep them alive. It's not true. It turns as we've talked about, it's turned out that the whole process with other genes can keep selecting the antimicrobials as we go along. The idea of this being a sort of fitness penalty, which meant that maintaining an antibiotic resistance gene was something that cost the organism, turns out it was evoked to explain why the resistance tended to drop off in a population of organisms over time, and why most of the genes are carried on these things called plasmids rather than being part of the main genome of the organisms themselves. But it turns out that what happens is that organisms with these genes can switch them off and still keep the genetic material, because it really doesn't, in the scheme of things, it doesn't cost them too much more to keep replicating those genes. But what it does do when they have the genes present is enable them to switch them on as soon as they discover that they are in an environment where there is something that is likely to cause them some sort of grief or reduce their competitive advantages. So the whole myth about fitness just doesn't fit anymore. The other one, and this was quite important, is that we thought that it would not be possible for genetic information to pass from, for example, enteric organisms, the organisms that like Salmonella, Shigella, E. coli, that cause and live in your gastrointestinal tract and cause diarrhea and those sorts of things. It was thought that it would be unlikely, even impossible, that their genetic information would wind up in organisms like staph or streptococcus, which are sort of more associated with the skin, respiratory tract, and so on, and are very different structurally. But it turns out that this is now widely accepted as being possible. It's even possible to exchange genes between bacteria and fungi, which was thought to be, almost impossible. But this is a process that's over the last 40 or so years has really evolved. It was actually part of the biotech boom where we learnt we could create a gene or synthesize some capability in, let's say, a laboratory organism like E. coli, but ultimately transfer it to something useful like a fungus that digested wood or something like that, or vice versa. So it's now a quite well-established process that you can transfer DNA and get it to stick across these wide generic and phylum differences.
You think we're going to get to a point where we realize we're transferring it just from people to people?
Eventually, I'll talk about that later on.
Okay.
This, I don't expect you to understand this whole diagram, but what it basically is saying that this is a graphical representation of that diverse transfer capability of this system, where normally speaking, I mean, somewhere in the middle of it is Escherichia, the standard E. coli of most lab work. Related organisms like Klebsiella. That's not surprising that these organisms can exchange genetic information. It was more surprising that you could get exchange between Escherichia and Pseudomonads. We've now established that it's relatively easy between that whole group of organisms, the purple ones there, and let's say the cocci, the Staphylococcus, Enterococcus, which are very different bugs. But what's actually causing us grief in hospitals at the moment is the group at the top, the Myroides and the Bacteroides, which we used to think were non-pathogenic, but they have become so, particularly in a hospital setting, by this process whereby genes can be transferred from the usual suspects, as we say, to these new organisms. And if anyone wants the reference, it's there on the slide. I can provide it later if you want. So that's just an example. So as we've talked about, one of the other myths was that DNA would not survive outside the cell. And anyone who's worked with DNA will certainly appreciate that that was not such a surprising conclusion in our early experiments, because it was extraordinarily difficult to isolate DNA and to work with it. It always seemed to be breaking down and being digested by other things. But it turns out that particularly in soil and certainly in wastewater as well, you've got this vast soup of genetic material. It's like a hardware store where microbes can acquire sort of pre-constructed genes that confer various capabilities on them as they need and when they need to survive in those environments. So it's now quite well established that DNA is free in soil in particular. And I've talked about the idea of transfer between phyla, which are sort of the very distinct families of microbes that you find. But the important thing is that in addition to antimicrobial resistance genes, there are what are called broad host range genes that enable those resistance genes to be transferred to and expressed in just about any species of microbe that's in that environment. And so this is a really important area where the organisms that can be the reservoirs of antimicrobial resistance DNA are not actually organisms that we would normally consider of importance or concern in, say, human health and medicine. So this is just one example of a number of things I've been talking about. This is a plasmid that's been isolated. I think it was isolated from a Bacteroides, but to show the kinds of determinants that are present. Don't worry about the names of the various genes, but the net effect is that the yellow areas are required, genetic information required for the replication of this whole piece of DNA, this plasmid. The brown areas are related to the ways in which it can be transferred from one cell to another. Blue is about whether or not genes are transferred into the chromosome of the host organism or just kept on the plasmid. But then and it's so is a sort of orangey brown at the bottom there. But the crunch comes in the pink area, which is nearly half of this particular structure. And there must be 6 or 8 different resistance genes carried in that stretch. And there are also mechanisms whereby additional ones can be added without any great difficulty. So this is a sort of a Swiss Army knife plasmid that enables organisms to survive in all sorts of contexts and resist antimicrobial agents, but also enables them to transfer between different organisms. So I don't think there's any argument now that there are antimicrobial resistant microbes and AMR DNA in soil. But what does that mean? And the short of it is that there's not yet much consensus, although it's changing and developing. The important thing, I suppose, is that we don't have confirmed cases where resistant infections have definitively been acquired from soil. And part of the reason for that is that, as I pointed out earlier, there is a delay between, say, us as workers in soil being exposed and the person getting sick as a result of those organisms to which we are exposed and were infected by. It can take days or weeks for that process to happen, maybe even longer if people become what are called silent carriers. So we don't know for sure. We don't have a clear example that this is happening. It's interesting that we understand how chemical contaminants behave, but we have very little understanding of how microbiological contaminants behave in the same environment. And at the moment, sad to say that there's not a huge amount of awareness amongst us as professionals in this area, nor amongst the medical profession, who are still largely treating AMR as a hospital problem. It isn't. Try the next slide. It's got some regulatory implications. And you might ask, well, if this is a problem, why aren't there regulations about it? And why don't we have criteria? Well, ain't that simple? Part of it is that how do you set a criterion for something that multiplies? Chemical contaminants don't do that. So you can say, right, 2 micrograms of PCB per kilogram is your limit. But you can't say that about microbes because they multiply. There may be a threshold, but part of the problem is that because different people differ in their susceptibility to an infection or whatever, it's very difficult to say, right, this is your limit, and less than that, we're safe. More than that, there's a risk. And as I've pointed out, how do you assess the risk when the person exposed may not be the ultimate receptor? And how do you assess a risk anyway when we don't understand the dose response curve? For chemical contaminations, there's usually a well-established dose response curve. This much material will cause this effect. You can't say that about microbes, unfortunately. So there are some real problems with coming to terms with how we might regulate AMR. Let's go to part 3, Richard. What can we do as practitioners? Well, in the absence of all those criteria and guidance, about the best we can probably do at the moment is to stick with a combination of the techniques we use for PFAS and sterile technique that's used in hospitals. And basically what it means is to try and avoid meticulously contact with between your skin and your clothing, if you can, and soil or water. That's the first step in minimising the risk that you would acquire antimicrobial resistant organisms from soil. Using hand sanitizer is also probably warranted, but how many of us would realise that we've probably also got to disinfect our boots and those sorts of things? And so basically meticulous cleanliness throughout fieldwork is going to be important. But it's also important to remember that we're talking usually billions of organisms in a gram of soil. So it's very easy for an infection to occur. So say it usually doesn't make us sick, but it might make others. And what action should wastewater treatment adopt? Clearly, leaking or inadequate wastewater management systems are going to be a problem. But do the plants themselves have to take action? And even then, there are some problems. What do we look for? What protective measures do the individuals who are doing the monitoring have to wear? That sort of stuff. But what indicators should a wastewater treatment plant be looking for? And then how do we handle the samples? Those sorts of things. I don't have those answers yet, but they're certainly things that we have to consider.
Thanks for that, Brent. Plenty of food for thought. Yeah, really interesting. I think we'll go to the early bird questions. So we're running a little bit tight for time. So the first one, are there any emerging wastewater treatment options that could potentially solve this problem being researched?
Oh, I mean, there are numbers, but it all becomes, it boils down to the cost. I mean, ozone treatment, UV, will all help to basically sterilise the effluent. But can we afford to do that? One of the other ways is to look at ways of removing nucleic acids from wastewater streams, and that's really only becoming, only beginning. But there are probably some simple ways that could be done. Some of them relying on the fact that DNA, for example, sticks to hydroxylapatite, which is a phosphate mineral, and is removed very effectively by that. But DNA is also a charged molecule, so passing the effluent through a sort of an electrolytic system may well be a way of doing it. So it's in early stages yet, and this is because we haven't really appreciated that this might be a problem.
All right. Question number 2. How can we test/measure the presence of resistant pathogens? I'm keen to understand how we quantify this risk and its sources.
That's easy in the sense that the detection of resistant pathogens in a clinical context is bread and butter stuff, and you could adopt the same technologies, but it's not necessarily the best way to go. As I mentioned, many of the organisms that we maybe should be interested in are not culturable. They don't grow very well, and they may not grow very well at all after they've been through a sewage treatment plant, for example. But we can detect their DNA, and there are now quite simple and straightforward processes that for example, are called qPCR, quantitative PCR, or eDNA, environmental DNA, that would detect the presence of these organisms. So that's certainly possible. And to answer a couple of other questions that are like this, that testing is available if you ask for it. But the question of are we testing for the right things and in the right way is still open.
So, is it fair to say there's always resistant pathogens around, like you've spoken about trillions of cells and things? So you do your eDNA analysis or whatever, you do your DNA analysis, and you identify that. I mean, big whoop, it's there all the time. So is there a certain sort of percentage of cells where it becomes the issue?
But
Is
that's
that
the problem. That's exactly the problem. It's probably there, but what does it mean? And this gets back to that point I made earlier about setting criteria. We just don't know at this stage. We don't know enough about. We don't even know the answer to that. Your first question is it there? But once we do, there's not enough information yet to allow us to say, right, if this much is there, that's a problem and we need to do this. So we're at those early stages yet where we don't quite know how much resistant AMR DNA actually passes through our systems.
so? This question's a bit the same. What testing is available?
Yeah.
Therefore, overflows of pump stations and sewage treatment plants which discharge to recreation areas or irrigation water supply.
Yeah, I mean the likelihood is that each of those is actually discharging some antimicrobial resistant organisms and AMR DNA. What that means in terms of risk is difficult to know, beyond saying, I suppose, that we are not overwhelmed yet with infections by AMR organisms, which means there must be some degree of success in minimising these things in these emissions.
Do you think it's got anything to do with the way we treat it, or is it more just that there's lots of good guy microbes out there competing with the AMR microbes?
It's all of the above. That we have evolved over many years a quite effective wastewater management system if it's in place. And then we are helped by various organisms that will outcompete AMR organisms in the absence of, significant stressors and sort of things that promote competition. But it's going to be there. And it's that awareness that I guess we've got to talk about.
So we may need to reconsider the ways we currently manage wastewater, the current way and the new way to manage wastewater.
We certainly need to begin asking questions about how the way we manage wastewater could be improved to minimise AMR one way or another. And that's a fairly big question and a fairly extensive set of requirements on that industry. But it's likely that we are going to need to improve the way we manage wastewater.
Who's working on that?
There are a couple of groups. Sorry, more than that even. Off the top of my head, there's SAFE in South Australia. It's a CRC for the I can't remember what the SAFE stands for. Anyway, they are looking to try and minimise the impact of AMR across agriculture in particular. There's a group in CSIRO that's trying to address antimicrobial resistance from a One Health perspective, which is basically that all living things are potentially susceptible to AMR one way or another, and therefore that needs to be addressed. There's a group at University of Queensland, I think another one at QUT, and there are academics at University of Sydney and at Melbourne University that are working on these things as well.
Yeah. Next question. Are treated water discharges, e.g. burst water mains, impacting creek health, including macroinvertebrates?
I doubt that burst water mains of themselves would affect creek health and macroinvertebrates, because treated water, as in burst water mains, we're talking potable water. It will be burst sewers that would be the ones that I would be concerned about, and most wastewater treatment plants discharge to creeks and that sort of stuff directly anyway. So the issue of burst pipes thereafter is less of a problem, provided your effluent meets the appropriate standards. But yeah, the risk is certainly that things like macroinvertebrates would serve as hosts to organisms that carry AMR genes, etc.
Okay, question 6. How much does landfill leachate contribute to AMR, and are current irrigation or treatment methods enough to manage the risk?
Okay, short answer is I'm not sure to what extent AMR survives in landfill leachate. I suspect there are people who are looking into this, but I would consider leachate to be a much more challenging environment for most microbes than sewage is, for example. So it may be that the microbial load in leachate is much lower. That may not be the case for a putrescible landfill. I'm not entirely sure. But Yeah, I think I'd have to say I'm not sure how much is known.
I thought it's microbially heavy. It's coming out of effectively like a bioreactor.
Yeah, but you're also talking relatively extreme pH and that sort of stuff. So, as I say, it may not be an environment that they survive in. Um, but again, as you say, there's anywhere where there are lots of microbes, there are likely to be lots of AMR genes, um, even if they're not in species that we can either cultivate or that cause us problems.
All right, question number 7. Have you considered flood and mass water event impacts, downstream impacts from wastewater treatment plants and mold assessment techniques?
The short answer is no, because that wasn't my focus. But when you talk about mold, for example, the important point to remember is that fungi are able to acquire genes for antimicrobial resistance and for various other environmental stressors. Um, but yeah, I can't speak um, to the impacts downstream from wastewater treatment plants.
Last of the early bird questions. What are the relative risks of wastewater overflow during storm events versus treated wastewater discharge ongoing?
I think it's fair to say that overflow from a wastewater treatment plant will always contain more risk in terms of AMR than um, the treated ongoing discharge, simply because the treatment hasn't finished. You've got, when storm events cause the process to overflow, it's almost certain that the overflow will not meet the discharge criteria that are normally applied to these things. So in that sort of circumstance, you'd expect more organisms, more genetic material, and indeed quite possibly more antibiotics and that sort of stuff, because they haven't been broken down by the treatment process.
Okay, we're now going to switch to the Q&A. Got 10 questions to go. You
Oh,
happy to
goodness.
keep going?
I'm happy to keep going, but
All right. Well, there's still plenty of people here, Brent, so you're going to keep going. The first question, do we know which contaminants increase AMR risk specifically, and can we quantify the likely risks?
unfortunately, no, in the sense that, like, if I'm right in thinking that what you're saying is that if we knew there was X milligrams per kilogram of zinc available in a soil, would that lead to certain amounts of AMR? Um, that's about as precise as I could get. Again, organisms like us, or sort of microorganisms rather, like us, are very varied in their susceptibility and so on. So the idea of using if you like, an environmental indicator, like, let's say zinc or cadmium or something like that, the best you can know is that it is likely to select for resistant organisms one way or another. And certainly the presence of antibiotics will do that. But the relationship between the concentration of those agents and the um, the outcome in terms of resistant organisms is just not known, I think, at this stage.
Okay, next question by Kim Govan. How many known AMR genes are there?
Um, there are about 70 classes of clinically relevant antimicrobial agents, all of which are subject to resistance genes. And very often there are families of genes. So there might be 5 or 6 different types of gene that confer resistance to one or another of the penicillins, for example. So there's literally, in terms of individual genes, there might be thousands, but there will be common DNA sequences that we can look for. If you know that's one of the implications of that question, that we could probably find sequences that we could use for diagnostic and detection purposes for most of them.
Do they do that in hospitals now?
I think so. I think it's certainly starting.
Yeah.
It's often quicker than trying to cultivate, culture organisms, etc. PCR is now so quick and effective.
So Claire Elton's got a question. Antibiotics are created from microorganisms. So like in the past, won't we take these new microorganisms and make improved antibiotics? Or will they change too fast for us to keep up?
Yep, that's simple. The sad truth is that while soil microbes and microbes in wastewater will or can produce new antimicrobial agents, a fair proportion of them, most of them, are probably almost as lethal to people as they are to microbes. And so that's been one of the problems, that of all the potential antimicrobial agents that you can find in soil, very few of them are actually suitable for therapeutic use in mammals. So there are problems there. And then by the very fact that it is a natural substance, or even a derivative of a natural substance, there inevitably will already be something that leads to resistance out there. Like, you could find penicillin resistant organisms in the same soil from which Florey got his fungus, that made penicillin. So resistance is already out there, folks.
Has always been.
Yeah.
Next question. Is the suggestion that wastewater treatment becomes an amplifier for potential to generate AMR pathogens?
Certainly in some circumstances, yes. That point I made, that you only need a few bugs to become resistant, and they can then multiply in the presence of the selective agent. That's the difficulty with this whole AMR story, that you are talking about if you like, a contaminant that multiplies rather than stays constant or is depleted naturally, as chemical contaminants are.
All right, next question, another question from Kim Govan. Are there studies where a AMR gene was tagged and traced through the waste treatment plant process?
I'm pretty certain there are. I have accumulated a library of several thousand papers in this area, and I know I've seen one somewhere. But that's a fairly straightforward project experimentally, so I'm pretty sure it's been done.
So, Kim, if we find one, we'll shoot you
Yeah,
out an email.
I'll see what I can find.
That'd be great. Thanks, Brent. Next question's from John Franklin. Are secondary treatment wastewater treatment systems, brackets aerated wastewater treatment systems, which include tertiary treatment through chlorination or UV, effective at limiting potential transport of AMR and related genetic material into soil?
They're certainly effective at reducing, I wouldn't say totally preventing, but certainly reducing viable organisms. What hasn't been looked at in enough depth is whether these processes are sufficient to completely remove functional, intact AMR genes. In other words, DNA is not usually on the list of things you check when you check how effective your ozone treatment, tertiary water treatment is, for example. But I would expect ozone in particular would be fairly effective at breaking down DNA. But is it effective enough? I don't know.
Okay, next questions from Nicholas Mason. Have any studies been done in the natural attenuation rates of AMR in normal soil conditions or with exposure to sunlight?
The concept of natural attenuation is very different from what we would see with chemical contaminants. As I mentioned, the thing is that organisms will switch off their capability in the absence of a stressor. Sunlight and solar UV, etc., are actually stressors that can also be, there are genes that confer ability to tolerate those better than others. So yeah, it's too much of a movable feast, I suspect, for us to be able to predict natural attenuation rates and at what point natural attenuation is enough. You only need one bug to be present and the right stressor to be there for, as I say, a whole population to develop overnight.
Interesting looking at those predictions you had at the beginning of your presentation about, 10 million deaths by 2050,
2050,
and
yeah.
that was a cheery way to start. But I
True.
just wondered, like someone's obviously worked it out, right? They've said, okay, right now we have X number,
Mm.
and
Uh,
by then we'll have Y. I mean,
there's a whole heap of stuff produced by the World Health Organization who've actually declared that this is one of the top 10 health concerns on the planet. And that happened, they made that declaration about 2015. So it's been around a while. But as I said, the part of the difficulty is that people have said, oh, yeah, that's a clinical problem. That's a problem to do with hospitals and doctors and that sort of stuff. What they haven't appreciated until more recently is that there are all these processes in soil and that we ordinary soil scientists can wind up being the carriers, the vectors of this thing called AMR. That's a cheery thought.
yeah, I don't like where you're getting there. Don't like to think of what your cleanup option is. Uh, so Nicholas Mason's got a second part to his question. Secondly, what analytical and testing options for AMR is there for soil and water? And are they NATA accredited for robust assessment, including QA requirements? Or does that currently sit in a grey area without standardized methodologies?
Um, I don't know. They're entirely capable of being NATA accredited, but I haven't checked. I haven't actually used any such services myself, but they are certainly capable of being accredited. They're relatively well-established research methods involving PCR, either qPCR or eDNA, which is a version of PCR. I don't think they're commercially available, or they might be provided by one or two labs that aren't the standard analytical chemistry labs. But I also know that the chemistry labs are starting to scratch their heads very seriously about this area as well. So it won't be long.
I like this next question. It's from an anonymous attendee. Is it plausible to assess risk using indicators in a similar manner to that the way that E. coli is measured as an indicator of pathogen risk?
Short answer is probably yes. But the difficulty you have is that we have a large body of evidence and experience that says if you've got 100 E. coli cells, colony forming units per gram of soil, this is usually an indicator that there are likely to be sewage-derived pathogens present. That's just based on a lot of past work. We could possibly get there using DNA as well, but I don't believe that work has started. But it's certainly, I think, a possibility.
We're certainly getting there with those eDNA techniques,
Yeah.
And they're getting lower in cost.
Mm.
Jean Meeklam. Hello, Jean.
Hello,
Thanks,
Jean.
Brent. Excellent presentation. There you go. Very well explained. How do we manage the tension between good microorganisms that we in the soil ecosystem needs versus the AMR microbes?
If only I knew the answer to that. What is likely to help is minimizing the transport of what I've called stressors, be they antibiotics or the particularly the inorganic contaminants, the metals, into soil environments. That will certainly help. But you've got to say that where there's a risk that antibiotics will be present in an effluent, that pressure will always select for at least some resistant organisms. Whether they can be outcompeted by the good ones is an entirely different question, and just I don't think it's known, Gene. I really don't.
Right, last question before we let you off the hook. Kim Govan, one more question. Is methylation process known on-off switch in bacteria related to soil contaminants?
There is a process called methylation that happens inside cells that does switch on and switch off genes. It's one of many that can be utilized by organisms to manage the expression of their antimicrobial resistance genes. There are other simpler ones that are simply like the structural way that the DNA is arranged, whether it's carried by plasmids or not, and those sorts of things. Methylation, I'd put a fair way down the list in terms of the armory that organisms have to handle the stresses in their environment. But I think it does occur.
Brent, well, that brings us to an end of our questions. Thank you so much for today's presentation. It's fascinating and a little bit scary,
Yeah.
but really appreciate your contribution, and thanks for coming and doing a webinar again.
Oh, my pleasure. It's always a pleasure to work with you guys. I have great fun. Thank you for the opportunity.
Thanks, Brent. Thanks, everybody.
See you later. Bye for
Cheers.
now.